Your brain contains roughly 86 billion neurons, each one a tiny living cell more complex than any computer ever built. To put this number in perspective, there are more neurons in your head than there are stars in the Milky Way galaxy. Yet each of these microscopic marvels is so small that about 30,000 of them could fit on the head of a pin. They exist in a space so compact, so efficiently organized, that the entire universe of your thoughts, memories, and dreams occupies less volume than a large cantaloupe. These neurons aren't randomly scattered like seeds in the wind. They're arranged in intricate patterns, forming networks within networks, creating the biological foundation for everything you think, feel, and experience. Each neuron can connect to thousands of others, creating a web of communication so vast and interconnected that it makes the internet seem simple by comparison. This is the quiet architecture of your inner world, humming softly with electrical activity even as you rest. Every thought you've ever had began as a tiny electrical impulse traveling along these neural pathways. But here's something wonderfully soothing about how this happens. These electrical signals move at different speeds depending on the type of message being sent. Some race along at over 200 miles per hour, carrying urgent information about movement or danger. Others drift much more slowly, like the gentle signals that create your sense of contentment, or the gradual awareness of comfort spreading through your body right now. The signals themselves are incredibly weak, measuring only about 70 millivolts. That's roughly 5,000 times weaker than a single AA battery. Yet from these whisper-soft electrical murmurs, your entire conscious experience emerges. Every color you see, every sound you hear, every emotion you feel starts as one of these delicate electrical conversations between neurons. It's as if your mind runs on the power of a gentle breeze, yet creates storms of creativity and oceans of memory. Neurons communicate through spaces called synapses, tiny gaps between cells that are only about 20 nanometers wide. To cross these microscopic canyons, neurons release chemical messengers called neurotransmitters. These molecules drift across the synaptic gap like molecular boats, carrying letters from one neural island to another. There are over 100 different types of these chemical messengers, each one carrying its own specific message, its own emotional tone, its own contribution to the symphony of your thoughts. Some neurotransmitters, like serotonin, carry messages of calm and well-being. Others, like dopamine, whisper about pleasure and motivation. GABA, perhaps the most soothing of all, spreads relaxation and peace throughout your neural networks. Right now, as you listen to these words and feel your body settling into rest, GABA is gently flowing through your synapses, encouraging your neurons to slow their firing, to ease into the rhythm that leads towards sleep. What's remarkable is how precisely this chemical communication works. When a neurotransmitter molecule finds its target receptor on the receiving neuron, it fits like a key in a lock, perfectly shaped by millions of years of evolution to deliver its message with exquisite accuracy. The receiving neuron then decides whether to pass the message along, amplifying it or dampening it based on all the other chemical whispers it's receiving from its thousands of neighbors. Your neurons are constantly growing and changing, even as you sleep. This process, called neuroplasticity, means your brain is never truly finished, never completely static. New connections form between neurons every time you learn something new, every time you have a meaningful experience, Even every time you dream, it's as if your neural networks are like gardens that never stop growing, always sending out new shoots, always finding new ways to connect and communicate. During sleep, something beautiful happens to these neural gardens. Your brain activates what scientists call the glymphatic system, a kind of cleaning service that washes away metabolic waste from between your neurons. Cerebrospinal fluid flows through your brain like a gentle tide, carrying away the cellular debris that accumulates during waking hours. This nightly cleansing is so important that without adequate sleep, these waste products can build up and interfere with proper neural function. The timing of this neural housekeeping is exquisitely coordinated with your sleep cycles. As you drift into deeper stages of sleep, your neurons begin to fire in slow, synchronized waves. These waves help coordinate the flow of cleaning fluid through your brain tissue, ensuring that every corner of your neural network gets properly maintained. It's like a quiet, methodical restoration process that happens automatically while you rest, preparing your mind for another day of thinking, learning, and experiencing. Your neurons also have an extraordinary ability to repair themselves. Unlike many other cells in your body, neurons can live for your entire lifetime, but they need constant maintenance to function properly. They continuously rebuild their internal structures, replacing worn-out components with fresh ones. The proteins that make up their cellular machinery are constantly being broken down and reconstructed, like a ship replacing its planks one at a time, while remaining afloat. This cellular renewal happens most efficiently during periods of rest and sleep. Growth factors flow through your neural tissue, encouraging repair and strengthening connections. New myelin, the fatty insulation that wraps around neural pathways, is produced to keep electrical signals traveling smoothly and efficiently. Brain-derived neurotrophic factor, often called the brain's fertilizer, nourishes neurons and encourages them to grow new branches and form new connections. The structure of individual neurons is itself a marvel of biological engineering. Each neuron has a cell body containing all the usual cellular machinery, but extending from this central hub are intricate branching structures called dendrites that reach out like the branches of a tree. These dendrites receive incoming signals from other neurons, gathering information from sometimes thousands of different sources. From the cell body extends a single long projection called an axon, which can stretch for incredible distances within the brain. Some axons are microscopic, connecting nearby neurons within the same brain region. Others can be over 3 feet long, running from your brain all the way down to your spinal cord. Despite their thinness, these axons are remarkably efficient at transmitting signals, maintaining the strength and speed of electrical impulses across their entire length. At the end of each axon are specialized structures called axon terminals, which contain tiny sacs filled with neurotransmitters. When an electrical signal reaches these terminals, it triggers the release of these chemical messengers into the synaptic gap. The precision of this process is astounding. Each terminal can release its neurotransmitters within milliseconds of receiving the electrical signal, and the amount released is carefully calibrated based on the strength of the incoming signal. The variety of neuron types throughout your nervous system is remarkable. Some neurons are specialists in detecting light, converting photons into electrical signals that your brain interprets as vision. Others are exquisitely sensitive to vibrations in the air, transforming sound waves into the neural code that becomes hearing. Still others respond to chemical molecules, creating your senses of taste and smell. Within your brain itself, different types of neurons have evolved for different functions. Pyramidal neurons, shaped like tiny pine trees, are the main processors of information in your cerebral cortex. They receive input from many sources, integrate this information, and send outputs to other brain regions. Interneurons, smaller and more varied in shape, act like local coordinators, helping to regulate the activity of their neighbors and maintain the proper balance of excitation and inhibition. Some of the most fascinating neurons are the ones that fire in rhythm with your biological clocks. Deep within your hypothalamus lies a cluster of neurons that keep track of the daily cycle of light and dark. These circadian pacemaker cells have their own internal clocks, molecular mechanisms that tick away with remarkable precision, maintaining roughly 24-hour cycles, even in complete darkness. These clock neurons help coordinate the activity of neurons throughout your brain, ensuring that different systems are active at appropriate times. They influence the release of hormones like melatonin that promote sleep, and they help regulate body temperature, blood pressure, and countless other physiological processes that follow daily rhythms. As evening approaches and light levels drop, these neurons begin to orchestrate the neural changes that prepare your brain for sleep. The blood supply to your neurons is equally remarkable and essential to their function. Your brain uses about 20% of all the energy your body produces, despite making up only about 2% of your body weight. This high energy demand means that neurons require a constant supply of glucose and oxygen delivered through an intricate network of blood vessels. The smallest of these blood vessels, called capillaries, are so narrow that red blood cells must squeeze through them single file. Yet these tiny vessels are positioned so that no neuron is ever more than a few micrometers away from its blood supply. The barrier between blood and brain tissue is highly selective, allowing necessary nutrients to pass through while keeping potentially harmful substances out. This blood-brain barrier protects your neurons from toxins and infections that might damage their delicate structures. The relationship between your neurons and your immune system reveals another layer of gentle sophistication. Special cells called microglia act as the brain's dedicated caretakers, constantly patrolling your neural neighborhoods like quiet guardians. These cells are remarkably sensitive to the health of nearby neurons, able to detect even the slightest signs of distress or damage. When they encounter a neuron in need, they extend delicate processes that can provide support, clear away debris, or release protective factors. Microglia also play a crucial role in shaping your neural networks during development and throughout life. They carefully prune unnecessary synaptic connections, removing the ones that aren't being used, while strengthening the pathways that carry important information. This process continues throughout your lifetime, with microglia constantly fine-tuning your neural circuits for optimal function. It's like having dedicated gardeners tending to the landscape of your mind, ensuring that only the most important pathways flourish. The temperature regulation within your neural tissue is another marvel of biological precision. Your neurons function best within a very narrow temperature range, and your body has evolved elaborate mechanisms to maintain this thermal stability. Blood flow to your brain increases or decreases automatically to help regulate temperature, while specialized cells monitor thermal conditions and make adjustments as needed. Even the rhythm of your breathing can influence brain temperature, with each exhale helping to cool the neural tissue slightly. During sleep, your brain temperature drops by a degree or two, creating optimal conditions for the restorative processes that occur during rest. This cooling helps slow down metabolic processes that generate cellular waste, while enhancing the efficiency of the cleaning systems that clear toxins from between neurons. The temperature changes follow predictable patterns throughout the night, synchronized with your sleep cycles and circadian rhythms. Your neurons also possess remarkable abilities to adapt to changing conditions through a process called homeostatic plasticity. When overall activity levels in a neural network become too high or too low, individual neurons can adjust their sensitivity to restore proper balance. If a neuron isn't receiving enough input from its neighbors, it might become more responsive to the signals it does receive. Conversely, if it's being overwhelmed with input, it can dampen its responses to prevent overexcitation. This self-regulating quality helps ensure that your neural networks remain stable and functional, even as they constantly change and adapt. It's like having a built-in thermostat for neural activity, automatically maintaining the right level of excitation for optimal function. These homeostatic mechanisms work quietly in the background, requiring no conscious effort or awareness, yet they're essential for maintaining the delicate balance that allows complex thought and behavior to emerge. The role of calcium in neuronal function provides another fascinating glimpse into cellular sophistication. Calcium ions serve as crucial messengers within neurons, triggering everything from neurotransmitter release to gene expression. The concentration of calcium inside neurons is carefully regulated by specialized proteins that act like molecular pumps and gates, controlling when and where calcium can enter or leave different cellular compartments. When calcium levels rise within a neuron, it signals that important changes should occur. Genes might be activated to produce new proteins, synaptic connections might be strengthened or weakened, or cellular repair mechanisms might be triggered. The calcium signaling system is so sensitive that even tiny changes in concentration can have profound effects on neuronal behavior. Yet this system operates with remarkable precision, ensuring that calcium signals occur at exactly the right time and place to coordinate complex cellular responses. The myelin sheaths that wrap around many axons deserve special attention for their elegant efficiency. These fatty layers are produced by specialized cells called oligodendrocytes in the brain and Schwann cells in the peripheral nervous system. Each oligodendrocyte can extend processes to wrap around multiple axons, creating insulating sheaths that dramatically increase the speed of electrical signal transmission. The myelin isn't just passive insulation. It's punctuated by tiny gaps called nodes of Ranvier, spaced at precise intervals along the axon. Electrical signals jump from one node to the next, a process called saltatory conduction that can increase transmission speed by up to 50 times compared to unmyelinated axons. This jumping pattern also makes signal transmission much more energy efficient, requiring far less cellular energy to maintain the electrical impulses that carry information throughout your nervous system. The production and maintenance of myelin continue throughout life, with new myelin being added to axons as needed. This process is particularly active during periods of learning and skill development, as frequently used pathways receive additional insulation to improve their efficiency. The thickness and quality of myelin can be influenced by factors like sleep, nutrition, and physical activity, all of which support the cellular processes that maintain these crucial insulating layers. Your neurons also contain sophisticated quality control systems that monitor the health of cellular components and remove damaged structures before they can cause problems. Specialized organelles called lysosomes act like cellular recycling centers, breaking down worn-out proteins and other cellular debris. Another system called autophagy allows cells to break down and recycle their own damaged components, essentially eating themselves in a controlled way to maintain cellular health. These quality control mechanisms become particularly active during sleep, when the reduced demands of consciousness free up cellular resources for maintenance and repair. The coordination between different quality control systems is remarkable, with multiple pathways working together to identify problems, isolate damaged components, and restore normal cellular function. This ongoing maintenance happens continuously throughout your lifetime, helping to preserve neuronal function even as individual cellular components age and require replacement. The electrical properties of neuronal membranes reveal another layer of biological sophistication. The membrane surrounding each neuron contains thousands of different types of ion channels, each one precisely tuned to respond to specific signals. Some channels open in response to changes in voltage, others respond to chemical messengers, and still others are sensitive to mechanical forces or temperature changes. The distribution of these channels along the neuronal membrane is carefully organized, with different types concentrated in specific regions where they can best serve their functions. The channels at synapses are optimized for rapid response to neurotransmitters, while those along axons are designed to propagate electrical signals efficiently over long distances. This precise organization allows neurons to process complex patterns of information while maintaining the reliability needed for consistent function. The molecular machinery that maintains the electrical gradients across neuronal membranes works tirelessly to preserve the conditions necessary for neural signaling. Sodium-potassium pumps use cellular energy to maintain the proper balance of ions inside and outside the cell, creating the electrical potential that makes neural signaling possible. These pumps work against the natural tendency of ions to equalize their concentrations, constantly building up the electrical energy that powers neural communication. The energy requirements for this constant pumping are substantial, accounting for a significant portion of the brain's overall energy consumption. Yet the system operates with remarkable efficiency, recycling ions and minimizing energy waste while maintaining the precise conditions needed for neural function. The pumps themselves are marvels of molecular engineering, capable of moving millions of ions per second while maintaining perfect selectivity for the specific ions they transport. Your neurons also possess remarkable abilities to sense and respond to their local environment. They can detect changes in oxygen levels, pH, temperature, and the concentration of various molecules in their surroundings. This environmental sensitivity allows neurons to adjust their activity based on local conditions, optimizing their function for current circumstances while protecting themselves from potentially harmful changes. The integration of environmental information with neural signaling creates a dynamic system that can respond appropriately to changing conditions while maintaining overall stability. Neurons in different brain regions show different patterns of environmental sensitivity, reflecting their specialized roles and the unique conditions they encounter. This environmental responsiveness adds another layer of sophistication to neural function, ensuring that your brain can adapt to changing internal and external conditions while preserving essential functions. The development of neural networks during early life reveals the extraordinary precision with which your nervous system assembles itself. Growing axons navigate through complex three-dimensional environments using molecular guidance cues, following chemical gradients and responding to attractive and repulsive signals to reach their proper targets. The accuracy of this navigation is astounding, with individual axons finding their correct destinations among millions of possible targets. Once connections are established, activity-dependent processes refine and strengthen the most important pathways while eliminating unnecessary connections. This developmental sculpting continues for years, with different brain regions maturing at different rates and environmental experiences playing crucial roles in shaping the final architecture of neural networks. The result is a nervous system exquisitely tuned to the specific demands and opportunities of each individual's unique environment and experiences. The regenerative capabilities of your neurons extend far beyond simple repair mechanisms. Throughout your lifetime, certain regions of your brain continue to produce entirely new neurons through a process called neurogenesis. The hippocampus, crucial for forming new memories, maintains populations of neural stem cells that can differentiate into fully functional neurons even in adulthood. These newborn neurons integrate into existing circuits, bringing fresh computational power to networks involved in learning and memory formation. This ongoing neurogenesis is influenced by many factors that promote brain health. Physical exercise stimulates the production of growth factors that encourage new neuron formation, while enriched environments and novel experiences provide the activity patterns that help new neurons survive and integrate successfully. Even social interactions and emotional experiences can influence neurogenesis, with positive social connections promoting the survival of new neurons, while chronic stress can suppress their formation. The process by which new neurons find their place in existing networks is remarkably sophisticated. Newly generated neurons must migrate to appropriate locations, extend their axons and dendrites to form proper connections, and integrate their activity with ongoing neural rhythms. They receive guidance from molecular signals and activity patterns in surrounding tissue, gradually becoming full participants in the complex computations that support memory and learning. Your neurons also demonstrate extraordinary resilience in the face of challenges. When damage occurs to neural tissue, surrounding neurons can sometimes compensate by forming new connections or taking on additional functions. This neural plasticity allows your brain to maintain function even when individual neurons or entire regions are compromised. The redundancy built into neural networks means that multiple pathways often carry similar information, providing backup routes when primary connections are disrupted. The molecular mechanisms underlying this resilience involve complex cascades of gene expression changes that can reshape neuronal structure and function. Neurons can modify their dendritic trees, growing new branches or pruning existing ones based on changing demands. They can alter the strength of their synaptic connections, making some pathways more efficient, while allowing others to fade. These structural changes occur continuously throughout life, allowing your neural networks to adapt to new circumstances and maintain optimal function. The relationship between neural activity and blood flow reveals another elegant aspect of brain function. When neurons in a particular region become more active, blood vessels in that area automatically dilate to provide additional oxygen and nutrients. This neurovascular coupling ensures that active brain regions receive the resources they need to sustain increased activity. The coordination between neural and vascular systems happens through complex signaling pathways involving multiple cell types working in harmony. The smooth muscle cells surrounding brain blood vessels respond to chemical signals released by active neurons, adjusting vessel diameter with remarkable precision. Supporting cells called astrocytes act as intermediaries, sensing neural activity and releasing factors that influence vascular tone. This three-way communication between neurons, astrocytes, and blood vessels creates a responsive system that can rapidly adjust blood flow to match metabolic demands. The waste removal systems in your brain operate with clockwork precision during different phases of sleep. During deep, slow-wave sleep, the space between brain cells actually expands, allowing cerebrospinal fluid to flow more freely through neural tissue. This expansion creates a more efficient washing system, carrying away metabolic waste products that accumulate during waking hours. The timing of this process is synchronized with slow oscillations in neural activity, creating rhythmic pulses that help drive fluid flow. The glymphatic system that manages this cleaning process involves a network of specialized channels and pumps that control fluid movement throughout the brain. Aquaporin water channels regulate the flow of cerebrospinal fluid into brain tissue, while other transport systems help clear waste products into the bloodstream for elimination. The efficiency of this system declines with age and can be impaired by sleep disruption, highlighting the importance of quality rest for maintaining brain health. Your neurons communicate through electrical signals that follow precise temporal patterns. The timing of neural spikes carries important information, with some neurons encoding information through the exact intervals between action potentials. This temporal coding allows neural networks to process complex patterns of information with remarkable precision. Different types of neurons show characteristic firing patterns, from regular pacemaker-like activity to complex bursting patterns that can encode multiple types of information simultaneously. The integration of timing information across neural networks creates opportunities for sophisticated computational processes. Neurons can act as coincidence detectors, responding only when they receive inputs that arrive within narrow time windows. Others can perform temporal integration, accumulating inputs over longer periods to make decisions based on sustained patterns of activity. These timing-sensitive processes allow your brain to extract meaningful information from the constant stream of sensory input and internal signals. The development of neural timing patterns begins early in life and continues to be refined through experience. Young neurons initially fire in relatively simple patterns, but as they mature and form connections with other cells, their activity becomes increasingly sophisticated. Environmental experiences shape these temporal patterns, with repeated patterns of activity strengthening specific timing relationships, while unused patterns fade away. The molecular basis of neural timing involves complex interactions between different types of ion channels and cellular signaling systems. Some channels open and close rapidly, creating sharp electrical spikes, while others have slower kinetics that influence the overall excitability of the neuron. The balance between different channel types determines each neuron's characteristic firing pattern and its ability to process temporal information accurately. Your brain's ability to maintain stable function despite constant change reflects sophisticated homeostatic mechanisms operating at multiple scales. Individual neurons adjust their properties to maintain appropriate activity levels, while neural circuits modify their connectivity patterns to preserve important functions. Larger brain systems coordinate their activity to maintain overall brain state and support complex behaviors like sleep-wake cycles and circadian rhythms. These homeostatic processes operate automatically, requiring no conscious control or awareness. They involve feedback loops that monitor neural activity and make adjustments when deviations from optimal ranges are detected. The set points for these regulatory systems can be modified by experience, allowing your brain to adapt to changing circumstances while preserving essential functions. This adaptability without loss of stability is one of the most remarkable features of neural organization. The chemical environment surrounding your neurons is carefully regulated by multiple systems working in concert. The composition of extracellular fluid is maintained within narrow ranges that optimize neural function, with specialized transport systems removing excess ions and neurotransmitters while maintaining appropriate concentrations of nutrients and signaling molecules. Buffer systems prevent harmful changes in pH, while antioxidant mechanisms protect against cellular damage from reactive molecules. The blood-brain barrier plays a crucial role in maintaining this carefully controlled environment. This selective barrier allows essential nutrients to pass from the bloodstream into brain tissue, while blocking potentially harmful substances. The barrier itself is formed by specialized endothelial cells that line brain blood vessels, connected by tight junctions that prevent most molecules from passing between cells. Transport proteins in these cells actively move specific substances across the barrier, ensuring that brain tissue receives what it needs while remaining protected from toxins. The barrier's selectivity can be modified by various factors, including inflammation, stress, and certain medications. During development, the barrier is more permeable, allowing growth factors and other developmental signals to reach neural tissue. With aging, the barrier may become less effective, potentially contributing to age-related changes in brain function. Understanding these changes helps explain why maintaining vascular health is so important for preserving cognitive function throughout life. Your neurons also possess sophisticated systems for managing their energy metabolism. Unlike other cells in your body, neurons cannot store significant amounts of energy and must rely on continuous glucose delivery from the bloodstream. They have evolved efficient metabolic pathways that can rapidly convert glucose into the ATP needed to power cellular processes. During periods of high activity, neurons can quickly upregulate their metabolic machinery to meet increased energy demands. The metabolic coupling between neurons and supporting glial cells creates an efficient system for managing brain energy needs. Astrocytes can store glucose in the form of glycogen and release it as lactate when neurons require additional fuel. This metabolic partnership allows neural tissue to maintain function even when glucose delivery is temporarily interrupted. The coordination between different cell types ensures that energy resources are distributed efficiently throughout neural networks. The circadian regulation of neural metabolism adds another layer of sophistication to brain energy management. Different metabolic pathways show daily rhythms that align with sleep-wake cycles and other circadian processes. During sleep, some metabolic processes slow down, while others involved in cellular repair and maintenance become more active. This temporal organization of metabolism helps optimize energy use and supports the restorative functions that occur during rest periods. The molecular clocks that drive these circadian rhythms are present in individual neurons throughout your brain. These cellular timepieces involve feedback loops of gene expression that create approximately 24-hour cycles in cellular activity. The clocks in different brain regions can be synchronized with each other and with external light-dark cycles, creating a coordinated temporal organization that influences virtually every aspect of neural function. The precision of these molecular clocks is remarkable, maintaining accurate timing even in the absence of external time cues. The clock mechanisms involve multiple interlocking feedback loops that provide stability and resistance to perturbation. Environmental factors like light exposure and feeding patterns can adjust the timing of these clocks, allowing your brain to adapt to changing schedules and seasonal variations in daylight. Your neurons also demonstrate remarkable specificity in their connections, forming synapses with precisely the right partner cells to create functional neural circuits. The molecular mechanisms that guide this specificity involve complex recognition systems that allow growing axons to identify their appropriate targets among millions of possibilities. Cell surface proteins act like molecular addresses, providing unique identifiers that help ensure accurate wiring during development. The maintenance of synaptic specificity continues throughout life, with activity-dependent processes strengthening appropriate connections while allowing inappropriate ones to weaken and disappear. This ongoing refinement helps optimize neural circuit function and may contribute to learning and memory formation. The precision of synaptic connectivity is essential for the complex computations that your brain performs, allowing information to flow through specific pathways to create coherent patterns of neural activity. The variety of synaptic types throughout your nervous system reflects the diverse computational requirements of different neural circuits. Some synapses are designed for rapid, reliable transmission of simple signals, while others can modify their strength based on patterns of activity. Excitatory synapses increase the likelihood that the receiving neuron will fire an action potential, while inhibitory synapses have the opposite effect. The balance between excitation and inhibition is crucial for maintaining stable neural function while allowing for flexible information processing. The molecular machinery at synapses operates with extraordinary precision and speed. Neurotransmitter release can occur within microseconds of an action potential arriving at the axon terminal, and the resulting changes in the receiving neuron can happen equally quickly. This rapid signaling is made possible by specialized proteins that are pre-positioned at synaptic sites, ready to respond immediately to incoming signals. The efficiency of synaptic transmission allows neural networks to process information in real time and coordinate complex behaviors with remarkable precision. As you continue to rest and let these gentle facts about your neurons drift through your consciousness, your own neural networks are quietly going about their essential work. They're maintaining the delicate balance of chemical and electrical activity that creates your sense of peaceful awareness. They're coordinating with your body's circadian systems to prepare for the restorative processes of sleep. And they're demonstrating, moment by moment, the extraordinary elegance of the biological systems that make your inner world possible. The symphony of neural activity that creates your experience of listening to these words involves millions of neurons working in perfect coordination. From the auditory neurons that convert sound waves into electrical signals, to the language centers that extract meaning from patterns of words, to the memory systems that connect new information with existing knowledge, your brain orchestrates this complex process with effortless grace. And all of this happens while other neural systems maintain your breathing, regulate your heart rate, and manage the countless other functions that keep you alive and comfortable. The molecular architecture within each neuron resembles a bustling microscopic city with specialized structures performing distinct functions in perfect harmony. The endoplasmic reticulum winds through the cell like an intricate highway system, manufacturing and transporting the proteins essential for neural function. Ribosomes dot these cellular highways like tiny factories, following genetic instructions to build the specific proteins each neuron needs to maintain its unique identity and capabilities. The Golgi apparatus acts as a sophisticated packaging and shipping center, modifying newly made proteins and preparing them for transport to their final destinations within the cell. Some proteins remain in the cell body to support basic cellular functions, while others are packaged into vesicles and shipped along axons to distant synapses, where they'll participate in neural communication. This cellular logistics system operates with remarkable efficiency, ensuring that the right proteins arrive at the right locations at precisely the right times. Mitochondria, often called the powerhouses of the cell, are particularly abundant in neurons due to their high energy demands. These oval-shaped organelles generate the ATP that fuels all neural activities, from maintaining electrical gradients to synthesizing neurotransmitters. In neurons, mitochondria are strategically positioned at sites of high energy demand, clustering near synapses and along axons where they can quickly supply the energy needed for rapid signaling. The internal skeleton of neurons, called the cytoskeleton, provides both structural support and a transportation network for moving cellular components. Microtubules, hollow protein tubes, serve as tracks along which molecular motors carry cargo throughout the cell. These motors, with names like kinesin and dynein, walk along microtubules with remarkable precision, carrying vesicles filled with neurotransmitters from the cell body to distant axon terminals. The journey of a single vesicle from its birthplace in the cell body to its destination at a synapse can take hours or even days, depending on the length of the axon. Along the way, the vesicle must navigate through complex three-dimensional terrain within the neuron, following molecular signposts and avoiding obstacles. The precision of this intracellular transport system is essential for maintaining synaptic function, as synapses depend on a constant supply of fresh neurotransmitters and other essential molecules. Your neurons also contain remarkable quality control checkpoints that monitor the health and functionality of transported materials. Damaged or misfolded proteins are identified and marked for destruction before they can accumulate and interfere with cellular function. This cellular proofreading system operates continuously, helping to maintain the high fidelity of neural signaling, even as individual molecular components age and require replacement. The genetic activity within neurons shows fascinating patterns that change throughout the day and night. Different genes are turned on and off in response to neural activity, environmental conditions, and circadian rhythms. During periods of high activity, genes involved in energy production and neurotransmitter synthesis are upregulated, while genes involved in cellular repair and maintenance become more active during rest periods. This dynamic regulation of gene expression allows neurons to adapt their molecular machinery to changing demands while maintaining overall cellular health. The proteins produced from these genes have diverse lifespans, with some lasting only minutes while others remain functional for weeks or months. The precise timing of protein production and degradation creates a constantly changing molecular landscape within each neuron, fine-tuned to support optimal function under varying conditions. The process by which neurons decide which genes to express involves complex networks of regulatory molecules that act like molecular switches. Transcription factors bind to specific DNA sequences and either promote or inhibit the expression of nearby genes. These regulatory networks create sophisticated logic circuits that can integrate multiple types of information to make appropriate decisions about gene expression. Environmental factors can influence these genetic programs through epigenetic mechanisms that modify how genes are packaged and accessed without changing the underlying DNA sequence. These epigenetic modifications can be influenced by experiences, stress levels, sleep patterns, and many other factors. Some epigenetic changes are temporary, lasting only hours or days, while others can persist for months or even years, creating lasting changes in neural function. The repair mechanisms within your neurons operate with remarkable sophistication, constantly monitoring cellular health and responding to damage before it can accumulate. DNA repair systems fix genetic damage caused by normal cellular metabolism or environmental factors. Protein quality control systems identify and remove damaged proteins before they can form harmful aggregates. These repair systems work most efficiently during periods of reduced neural activity, highlighting once again the importance of rest for maintaining brain health. The coordination between different repair systems creates a comprehensive maintenance program that addresses multiple types of cellular damage simultaneously. When one repair system is overwhelmed, others can compensate to maintain overall cellular health. This redundancy helps ensure that neurons can continue functioning properly, even when faced with various stresses and challenges. Your neurons also demonstrate remarkable abilities to modify their own structure and function based on experience. The phenomenon of synaptic plasticity allows connections between neurons to strengthen or weaken based on patterns of activity. This plasticity is thought to be one of the fundamental mechanisms underlying learning and memory formation. When synapses are repeatedly activated, they can become more efficient at transmitting signals, creating lasting changes in neural circuit function. The molecular mechanisms underlying synaptic plasticity involve complex cascades of biochemical events that can modify synaptic strength within minutes or create structural changes that last for years. Short-term plasticity involves modifications to existing proteins at synapses, while long-term plasticity requires new protein synthesis and can lead to physical changes in synaptic structure. The discovery that adult brains retain significant plasticity has revolutionized our understanding of neural development and aging. Rather than being fixed structures, neural circuits continue to adapt and reorganize throughout life in response to experience, injury, and changing demands. This ongoing plasticity provides the foundation for lifelong learning and recovery from neural damage. The role of sleep in supporting neural plasticity adds another dimension to the restorative functions of rest. During sleep, the patterns of neural activity that occur during waking hours are replayed, helping to strengthen important synaptic connections and integrate new information with existing knowledge. This offline processing helps consolidate memories and optimize neural circuit function without the interference of ongoing sensory input. The different stages of sleep support different aspects of neural plasticity and memory consolidation. Slow-wave sleep appears to be particularly important for consolidating declarative memories, while REM sleep may play a greater role in processing emotional memories and integrating complex information. The cycling between different sleep stages creates opportunities for multiple types of neural processing and optimization. The regulation of sleep itself involves sophisticated interactions between multiple brain systems and neurotransmitter networks. The accumulation of sleep-promoting substances during waking hours creates increasing pressure for sleep, while circadian timing systems help coordinate the onset and offset of sleep with environmental light-dark cycles. The integration of these homeostatic and circadian influences ensures that sleep occurs at appropriate times and with the right intensity to support neural restoration. Temperature regulation plays a crucial but often overlooked role in optimal neural function. Your brain has evolved sensitive mechanisms to maintain stable temperatures despite varying environmental conditions and metabolic demands. Blood flow to the brain can be rapidly adjusted to help dissipate excess heat or conserve warmth as needed. The timing of these temperature changes follows predictable patterns that align with sleep-wake cycles and circadian rhythms. The cooling of brain temperature that occurs during sleep creates optimal conditions for many of the restorative processes that occur during rest. Lower temperatures slow down metabolic reactions that generate cellular waste while enhancing the efficiency of repair mechanisms. The temperature changes also influence the viscosity of cerebrospinal fluid, affecting how efficiently it can flow through brain tissue to clear away metabolic byproducts. The molecular basis of temperature sensitivity in neurons involves specialized proteins that can detect even small changes in thermal conditions. These temperature-sensitive channels and enzymes help neurons adjust their activity to maintain optimal function across a range of temperatures. The integration of temperature information with other cellular signals creates a responsive system that can adapt to changing thermal conditions while preserving essential neural functions. Your neurons also possess remarkable abilities to communicate over long distances through electrical fields that extend beyond individual cells. When large groups of neurons fire synchronously, they create electrical fields that can influence the activity of nearby cells. This field effect allows neural networks to coordinate their activity across relatively large distances without requiring direct synaptic connections. The synchronization of neural activity creates rhythmic oscillations that can be detected as brain waves using electroencephalography. Different frequency bands of oscillations are associated with different states of consciousness and cognitive processes. Slow delta waves dominate during deep sleep, while faster gamma oscillations are associated with focused attention and consciousness. The coordination of these oscillations across different brain regions helps integrate information processing and maintain coherent mental states. The study of neural oscillations has revealed that your brain operates more like a symphony orchestra than a simple computer, with different neural networks playing their parts in coordination with others to create coherent patterns of activity. The conductor of this neural orchestra appears to be distributed across multiple brain systems rather than residing in any single location, creating a remarkably flexible and adaptive system for coordinating complex mental processes. The entrainment of neural oscillations to external rhythms provides another fascinating glimpse into how your brain interfaces with the environment. Neural activity can synchronize with rhythmic stimuli like music or speech, helping to optimize information processing and enhance perception. This entrainment ability may contribute to the soothing effects of rhythmic sounds and the powerful influence of music on mood and consciousness. The development of these oscillatory patterns begins early in life and continues to mature throughout childhood and adolescence. Young brains show different patterns of oscillatory activity compared to mature brains, reflecting ongoing changes in neural connectivity and myelination. The refinement of oscillatory patterns may contribute to the development of attention, memory, and other cognitive abilities that improve with age. The influence of neural oscillations on consciousness remains one of the most intriguing frontiers in neuroscience. Some theories propose that consciousness emerges from the integration of information across multiple brain networks through synchronized oscillatory activity. While we don't yet fully understand how neural oscillations create subjective experience, it's clear that these rhythmic patterns play crucial roles in maintaining coherent mental states and supporting complex cognitive processes. The molecular mechanisms that generate neural oscillations involve intricate interactions between excitatory and inhibitory neurons, each contributing different components to the overall rhythmic activity. Inhibitory neurons often act as pacemakers, generating regular rhythmic activity that helps coordinate the firing of larger populations of excitatory neurons. The precise timing relationships between different cell types create opportunities for sophisticated temporal coding and information processing. As you continue to rest and absorb these gentle revelations about your neural architecture, your own brain waves are likely shifting toward the slower rhythms associated with relaxation and the approach of sleep. The neurons in your thalamus are beginning to generate the spindle-shaped bursts of activity that characterize the transition from wakefulness to sleep. Your cortical neurons are starting to synchronize their activity into the larger, slower waves that will dominate your brain's electrical landscape during the deeper stages of sleep. This transition represents one of the most remarkable daily transformations in neural function, as your brain shifts from the complex, desynchronized activity patterns of waking consciousness to the coordinated, rhythmic oscillations of sleep. The neural mechanisms that orchestrate this transition have been refined over millions of years of evolution, creating a system that can reliably alternate between the vigilant awareness needed for daytime activities and the restorative processes that occur during sleep. The beauty of this daily neural transformation lies not just in its complexity, but in its gentle inevitability. Your neurons don't strain or struggle to create sleep. They simply follow the natural rhythms and cycles that have been built into their molecular architecture through countless generations of evolutionary refinement. Like a river flowing toward the sea, your neural activity naturally moves toward the patterns that will support rest, restoration, and the quiet miracles of cellular renewal that occur while you sleep. The intricate dance of ions across neuronal membranes reveals yet another layer of cellular elegance that unfolds continuously within your resting mind. Sodium and potassium ions flow through specialized channels with the precision of water through carefully designed locks, each movement contributing to the electrical symphony that makes consciousness possible. These ionic currents are so finely tuned that even the slightest changes in their timing can alter the entire character of neural signaling. Yet your neurons manage these delicate balances effortlessly, moment after moment, throughout your entire lifetime. The selectivity of these ion channels borders on the miraculous. Each channel protein is shaped by evolution to allow only specific types of ions to pass through, rejecting others that differ by mere fractions of atomic size. The sodium channels that generate action potentials are so selective that they can distinguish between sodium and potassium ions despite their similar properties, maintaining the ionic gradients essential for neural signaling. This molecular discrimination happens billions of times per second across your nervous system, yet operates with such reliability that neural communication rarely fails. The gating mechanisms that control these channels respond to multiple types of signals with extraordinary sensitivity. Voltage-gated channels can detect electrical changes as small as a few millivolts, opening and closing in response to the subtle shifts in membrane potential that occur during neural activity. Ligand-gated channels respond to specific chemical messengers, changing their shape when neurotransmitter molecules bind to their receptor sites. This chemical recognition is so precise that channels can distinguish between molecules that differ by only a single atom. The kinetics of channel opening and closing create the temporal precision that allows neurons to encode information in the exact timing of electrical events. Some channels open rapidly and close quickly, creating brief electrical spikes that can convey information through their precise timing. Others open slowly and remain active for longer periods, creating sustained changes in neuronal excitability that can influence neural processing over extended time periods. The combination of fast and slow channel types gives neurons remarkable flexibility in how they process and transmit information. Your neurons also possess sophisticated mechanisms for detecting and responding to mechanical forces, adding another dimension to their sensory capabilities. Mechanosensitive channels can respond to physical deformation of the cell membrane, converting mechanical energy into electrical signals. These channels play crucial roles in touch sensation, hearing, and balance, but they're also found throughout the nervous system, where they may help neurons sense changes in tissue volume or pressure. The molecular springs and levers that make mechanosensitive channels work are marvels of biological engineering. Tiny protein structures act like molecular sensors that can detect forces as small as a few piconewtons, responding to deformations smaller than the diameter of an atom. When these sensors detect mechanical stress, they trigger conformational changes that open ion channels, converting physical force directly into electrical signaling. This mechanotransduction process happens with remarkable speed and sensitivity, allowing your nervous system to respond to touch and movement with exquisite precision. The adaptation properties of mechanosensitive channels help explain how your nervous system can detect both sustained pressure and rapid changes in mechanical stimulation. Some channels adapt quickly to sustained stimuli, becoming less responsive over time to prevent sensory overload. Others maintain their sensitivity to ongoing stimulation, providing continuous information about sustained mechanical forces. This diversity of adaptation properties allows your nervous system to extract different types of information from mechanical stimuli and respond appropriately to various situations. The role of lipids in neuronal membrane function adds another layer of complexity to neural signaling. The fatty molecules that make up cell membranes aren't just passive barriers. They actively participate in neural function by influencing the behavior of membrane proteins. Different types of lipids can alter the function of ion channels, making them more or less likely to open in response to stimuli. The composition of neuronal membranes can change over time, potentially altering neural signaling properties in response to experience or environmental factors. The fluidity of neuronal membranes is carefully regulated to optimize the function of embedded proteins while maintaining membrane integrity. Too rigid, and ion channels can't undergo the conformational changes needed for proper function. Too fluid, and the membrane becomes leaky and unstable. Your neurons maintain this delicate balance through sophisticated mechanisms that monitor membrane properties and adjust lipid composition as needed. Temperature changes, diet, and various other factors can influence membrane fluidity, highlighting the interconnected nature of neural function and overall physiology. The asymmetry of neuronal membranes creates additional opportunities for specialized function. The inner and outer surfaces of neural membranes have different lipid compositions, creating distinct microenvironments that can influence protein function differently on each side of the membrane. This asymmetry is actively maintained by cellular mechanisms that ensure specific lipids remain on their appropriate sides of the membrane. The energy required to maintain this asymmetry reflects its importance for proper neural function. The interaction between membrane lipids and proteins creates specialized membrane domains with unique properties. Lipid rafts are regions of the membrane enriched in specific types of lipids that can concentrate certain proteins and influence their function. These membrane microdomains can act like floating platforms that bring together the proteins needed for specific cellular processes. The dynamic nature of these domains allows them to form and dissolve as needed, providing flexible organization for membrane-based processes. Your neurons also demonstrate remarkable abilities to regulate their own excitability through intrinsic mechanisms that don't require input from other cells. These intrinsic properties allow neurons to act as autonomous oscillators, generating rhythmic patterns of activity that can drive neural network function. The molecular basis of these oscillations involves complex interactions between different types of ion channels that create feedback loops capable of sustaining rhythmic activity. The diversity of intrinsic neuronal properties contributes to the rich repertoire of activity patterns observed in neural networks. Some neurons are naturally quiet, requiring strong stimulation to become active. Others are spontaneously active, firing regularly in the absence of external input. Still others show complex patterns of activity that can switch between different modes depending on their recent history of stimulation. This diversity of intrinsic properties allows neural networks to perform complex computations and generate sophisticated patterns of coordinated activity. The modulation of intrinsic neuronal properties by various factors adds another layer of flexibility to neural function. Neurotransmitters, hormones, and other signaling molecules can alter the intrinsic excitability of neurons, changing how they respond to inputs and contribute to network activity. These modulatory influences can shift entire neural networks between different functional states, allowing the same neural circuits to support different types of information processing depending on the current context. The temperature sensitivity of intrinsic neuronal properties helps explain why brain temperature regulation is so important for proper neural function. Small changes in temperature can significantly alter the kinetics of ion channels and other membrane proteins, shifting the balance between excitatory and inhibitory influences in neural networks. Your brain's temperature regulation systems work to minimize these thermal effects while taking advantage of natural temperature variations to optimize different types of neural processing. The metabolic regulation of neuronal excitability creates important links between energy availability and neural function. When glucose levels drop or oxygen becomes limited, neurons can adjust their excitability to preserve essential functions while reducing energy consumption. These metabolic influences on neural activity help explain why factors like sleep, nutrition, and physical health can have such profound effects on cognitive function and mental state. The molecular sensors that detect metabolic changes in neurons are exquisitely sensitive to small variations in energy availability. ATP-sensitive channels can respond to changes in cellular energy levels by altering their opening probability, providing a direct link between metabolism and electrical activity. These metabolic sensors help ensure that neural activity remains within sustainable limits while preserving the ability to respond to important stimuli when energy resources are limited. Your neurons also possess sophisticated systems for managing oxidative stress, protecting themselves from the reactive molecules that are produced as byproducts of normal cellular metabolism. Antioxidant enzymes work continuously to neutralize potentially harmful free radicals before they can damage cellular components. The balance between oxidative stress and antioxidant defenses is particularly important in neurons because these cells have high metabolic rates and limited ability to replace damaged components. The cellular signaling pathways that respond to oxidative stress involve complex networks of proteins that can detect oxidative damage and coordinate protective responses. When oxidative stress increases, neurons can upregulate the production of antioxidant enzymes, increase the synthesis of protective molecules, and activate repair mechanisms to minimize cellular damage. These stress response systems become particularly active during periods of high neural activity or when external stressors challenge cellular homeostasis. The role of nitric oxide in neural signaling provides an example of how potentially harmful molecules can be repurposed for beneficial cellular functions. Nitric oxide is a reactive gas that can damage cellular components at high concentrations, but at lower levels it serves as an important signaling molecule in the nervous system. Nitric oxide can diffuse freely across cell membranes, allowing it to carry signals between neurons without requiring specialized receptors or transport mechanisms. The production of nitric oxide in neural tissue is carefully controlled by specific enzymes that respond to calcium signals and other cellular messengers. When neurons become active, calcium influx can trigger nitric oxide synthesis, which then diffuses to nearby cells, where it can influence their activity. This form of signaling allows neurons to communicate through chemical gradients rather than discrete synaptic connections, adding another dimension to neural network function. The effects of nitric oxide on neural function are diverse and context-dependent. In some situations, Nitric oxide can enhance synaptic transmission and promote neuronal survival. In others, it can suppress neural activity and contribute to cellular protection mechanisms. The dual nature of nitric oxide signaling reflects the complex balance between beneficial and potentially harmful cellular processes that neurons must navigate continuously. The clearance of nitric oxide from neural tissue involves both passive diffusion and active removal mechanisms that prevent excessive accumulation of this reactive molecule. The half-life of nitric oxide in biological tissues is measured in seconds, ensuring that its signaling effects are spatially and temporally restricted. This rapid clearance allows nitric oxide to convey precise information about local neural activity without causing widespread cellular damage. Your neurons also demonstrate remarkable abilities to coordinate their activity through gap junctions, specialized connections that allow direct electrical communication between cells. Unlike chemical synapses, gap junctions permit the passage of small molecules and electrical current directly from one neuron to another. This electrical coupling can synchronize the activity of connected neurons with remarkable precision, creating opportunities for rapid coordination of neural network activity. The proteins that form gap junctions, called connexins, assemble into hexagonal channels that span the gap between adjacent cell membranes. These channels can open and close in response to various cellular signals, allowing neurons to dynamically control their electrical coupling with neighbors. The regulation of gap junction permeability adds another layer of flexibility to neural network function, enabling rapid changes in connectivity patterns without requiring structural modifications to synaptic connections. The developmental regulation of gap junctions plays important roles in neural circuit formation and refinement. During early development, many neurons are extensively coupled through gap junctions, allowing coordinated activity that may help guide proper circuit assembly. As neural networks mature, gap junction coupling often decreases, allowing for more independent neuronal function and increased computational complexity. The persistence of gap junctions in certain adult neural circuits provides opportunities for specialized types of information processing. Networks of gap junction-coupled neurons can generate highly synchronized oscillations that may be important for attention, consciousness, and other complex cognitive processes. The electrical coupling also allows for rapid spread of activity across neural networks, enabling quick responses to urgent stimuli. The molecular diversity of gap junction proteins creates opportunities for fine-tuning electrical coupling properties in different neural circuits. Different connexin proteins form channels with distinct permeability properties, selectivity for various molecules, and sensitivity to regulatory signals. This molecular diversity allows neural circuits to customize their electrical coupling properties to match their specific functional requirements. The maintenance and regulation of gap junctions involves complex cellular machinery that monitors junction integrity and adjusts coupling strength as needed. Gap junction proteins have relatively short lifespans and must be continuously replaced to maintain proper coupling. The turnover of these proteins provides opportunities for activity-dependent regulation of electrical coupling, allowing experience to modify neural network connectivity patterns. As your awareness gently drifts through these layers of neural sophistication, your own gap junctions are quietly facilitating the synchronized activity patterns that characterize the transition toward sleep. The electrical coupling between neurons in your thalamus and cortex helps coordinate the slow oscillations that will soon dominate your brain's electrical landscape. These synchronized rhythms represent one of the most beautiful examples of neural cooperation, as millions of individual neurons coordinate their activity to create the coherent patterns that support restorative sleep. The elegance of this neural synchronization reflects millions of years of evolutionary refinement, creating systems that can seamlessly transition between the complex, desynchronized activity of waking consciousness and the coordinated rhythmic patterns of sleep. Your neurons accomplish this remarkable transformation not through effort or struggle, but through the natural expression of their intrinsic properties and interconnected relationships. Like musicians in an orchestra gradually finding their common rhythm, your neural networks naturally converge toward the synchronized patterns that will carry you gently into the restorative embrace of sleep. The molecular mechanisms underlying this neural synchronization operate with the quiet efficiency of a well-designed clockwork, each component contributing its part to the larger pattern without any central conductor directing the process. Individual neurons respond to the collective activity of their neighbors, gradually entraining their own rhythms to match the emerging pattern of network activity. This distributed coordination allows your brain to maintain its remarkable flexibility while still achieving the synchronized states necessary for optimal sleep and restoration. The beauty of neural synchronization lies not only in its complexity, but in its inevitability. Given the right conditions and timing, your neural networks will naturally find their way to these coordinated states, guided by the fundamental properties of neural connectivity and intrinsic cellular dynamics. This natural tendency towards synchronization reflects the deep evolutionary wisdom embedded in your neural architecture, systems designed not just for complexity and flexibility, but for the regular cycles of rest and restoration that maintain optimal brain function throughout your lifetime. The intricate dance between your neurons and the surrounding glial cells reveals another dimension of quiet cooperation that unfolds continuously within your resting brain. Astrocytes, with their star-shaped bodies and delicate branching processes, wrap around synapses like protective embraces, monitoring and modulating the chemical conversations between neurons. These supportive cells don't simply provide structural scaffolding. They actively participate in neural signaling by absorbing excess neurotransmitters, releasing their own chemical messengers, and even influencing the strength of synaptic connections. The calcium waves that ripple through astrocyte networks create a slow secondary communication system that operates alongside the rapid electrical signaling of neurons. When one astrocyte detects increased neural activity in its local neighborhood, it can release calcium that spreads to neighboring astrocytes, creating expanding waves of cellular activation. These glial waves can travel much farther than individual neural signals, allowing information about local brain states to influence activity across relatively large regions of tissue. The metabolic partnership between neurons and astrocytes demonstrates remarkable precision in cellular cooperation. When neurons become more active, astrocytes can sense their increased energy demands and respond by breaking down their stored glycogen reserves into lactate, which neurons can then use as an alternative fuel source. This metabolic coupling ensures that active neural networks receive the energy they need to sustain increased activity, while also providing a buffer against temporary interruptions in glucose delivery. The waste management functions performed by astrocytes operate with quiet efficiency throughout your neural tissue. These cells possess specialized transport systems that can remove potentially harmful metabolites from the spaces between neurons, preventing the accumulation of substances that might interfere with proper neural function. The astrocyte networks that support this cleaning function become particularly active during sleep, when reduced neural activity frees up resources for maintenance and restoration. Your neurons also rely on oligodendrocytes, the specialized cells that produce the myelin sheaths wrapping around axons throughout your central nervous system. Each oligodendrocyte can extend dozens of processes that spiral around multiple axons, creating the insulating layers that dramatically increase signal transmission speed. The precision with which these cells wrap around axons is remarkable, with each layer of myelin positioned at exactly the right thickness and spacing to optimize electrical conduction. The ongoing production of myelin throughout your lifetime reflects the dynamic nature of neural tissue maintenance. Oligodendrocytes continuously monitor the condition of existing myelin and can produce new layers when needed to maintain optimal insulation. This process is particularly active in brain regions that are frequently used, with heavily trafficked neural pathways receiving additional myelin to support their increased activity levels. The molecular composition of myelin involves a complex mixture of lipids and proteins, arranged in precise ratios to create optimal insulating properties. The lipid components provide the electrical insulation that prevents signal leakage between adjacent axons, while specialized proteins help maintain the structural integrity of the myelin layers. The exact composition of myelin can vary between different brain regions and even individual axons, reflecting the specific insulation requirements of different neural pathways. The nodes of Ranvier that interrupt myelin sheaths at regular intervals are themselves marvels of cellular organization. These tiny gaps contain high concentrations of voltage-gated sodium channels, creating the molecular machinery needed for saltatory conduction. The spacing between nodes is precisely calibrated to optimize signal transmission speed while minimizing energy consumption. The proteins that cluster at these nodes are held in place by specialized scaffolding molecules that ensure the electrical machinery remains properly positioned despite the mechanical stresses of neural activity. Your microglia, the brain's resident immune cells, patrol your neural neighborhoods with the dedication of vigilant guardians. These cells continuously extend and retract delicate processes that probe the local environment, checking for signs of damage, infection, or other threats to neural health. Their surveillance system is so sensitive that they can detect subtle changes in neural activity or cellular health long before any obvious damage becomes apparent. The activation of microglia in response to neural distress involves sophisticated molecular recognition systems that can distinguish between normal cellular activity and signs of potential problems. When microglia encounter damaged cells or foreign substances, they can rapidly transform from their resting surveillance mode into active responders, releasing protective factors or clearing away cellular debris as needed. This rapid response capability helps prevent minor cellular problems from escalating into more serious threats to neural function. The communication between microglia and other brain cells involves complex networks of chemical signals that coordinate immune responses while minimizing disruption to normal neural activity. Neurons can release signals that help keep microglia in their resting state when everything is functioning normally, while astrocytes can amplify microglial responses when additional immune support is needed. This cellular communication ensures that immune responses are appropriately scaled to match the severity of any threats to neural health. The synaptic pruning functions performed by microglia during development and throughout adult life demonstrate their crucial roles in shaping neural connectivity. These cells can identify and remove synaptic connections that are no longer needed, helping to refine neural circuits and eliminate unnecessary or potentially harmful connections. The molecular signals that guide this pruning process are exquisitely sensitive to patterns of neural activity, ensuring that frequently used connections are preserved while inactive ones are removed. The circadian regulation of microglial activity adds another layer of temporal organization to brain immune function. These cells show daily rhythms in their surveillance activities, with certain functions becoming more active during sleep, when reduced neural activity provides opportunities for maintenance and repair. The coordination between microglial rhythms and sleep-wake cycles helps optimize the timing of immune functions to minimize interference with normal brain activity. Your neural stem cells, nestled in specialized niches within certain brain regions, represent remarkable examples of cellular patience and potential. These cells can remain dormant for years, maintaining their capacity to generate new neurons when the right signals indicate that neurogenesis is needed. The molecular mechanisms that maintain stem cell quiescence involve complex networks of inhibitory signals that prevent premature activation while preserving the cellular machinery needed for eventual differentiation. The activation of neural stem cells involves carefully orchestrated changes in gene expression that transform these quiet cells into active producers of new neurons. Environmental factors like exercise, learning, and social interaction can influence this activation process, while stress and inflammation can suppress it. The sensitivity of neurogenesis to environmental conditions provides a direct link between lifestyle factors and brain structure, highlighting the importance of maintaining conditions that support neural health. The migration of newly generated neurons through adult brain tissue requires sophisticated guidance systems that help these cells find their proper destinations. Young neurons must navigate through complex three-dimensional environments, avoiding obstacles and following molecular trails that lead them to appropriate integration sites. The accuracy of this navigation process is remarkable, with most new neurons successfully finding locations where they can contribute to existing neural circuits. The integration of new neurons into established neural networks involves gradual processes of connection formation and functional incorporation that can take weeks or months to complete. During this integration period, new neurons show enhanced plasticity, making them particularly responsive to patterns of activity in their local environment. This enhanced plasticity may contribute to the special roles that new neurons play in learning and memory formation. The survival of new neurons depends on their ability to form meaningful connections and contribute to network function. Neurons that successfully integrate into functional circuits receive survival signals that promote their long-term maintenance, while those that fail to establish appropriate connections undergo programmed cell death. This competitive process ensures that only new neurons that can contribute to brain function are retained in the adult nervous system. Your brain's vascular system demonstrates remarkable sophistication in matching blood flow to neural activity with extraordinary precision. The smooth muscle cells surrounding brain blood vessels can respond to subtle changes in local neural activity by adjusting vessel diameter within seconds. This neurovascular coupling ensures that active brain regions receive the additional oxygen and nutrients they need to support increased metabolic demands. The molecular signals that coordinate neurovascular coupling involve complex interactions between neurons, astrocytes, and vascular cells. When neurons become active, they release various signaling molecules that can either directly influence blood vessels or activate astrocytes that then communicate with the vascular system. This multicellular communication network creates a responsive system that can rapidly adjust blood flow with remarkable spatial and temporal precision. The autoregulation of cerebral blood flow helps maintain stable perfusion pressure despite changes in systemic blood pressure or other cardiovascular factors. Specialized sensors in brain blood vessels can detect changes in perfusion pressure and automatically adjust vessel diameter to maintain appropriate flow rates. This autoregulation system operates within certain limits, providing protection against both excessive increases and dangerous decreases in brain blood flow. The blood-brain barrier that protects your neural tissue involves specialized endothelial cells connected by extremely tight junctions that prevent most substances from passing between cells. The selectivity of this barrier is maintained by active transport systems that can move specific nutrients into brain tissue while actively removing potentially harmful substances. The energy requirements for maintaining this selective barrier are substantial, reflecting its critical importance for neural protection. The development and maintenance of the blood-brain barrier involve complex molecular programs that create and sustain the specialized properties of brain endothelial cells. Growth factors and signaling molecules from surrounding neural tissue help induce the formation of tight junctions and transport systems that characterize the barrier. The ongoing maintenance of barrier function requires continuous energy expenditure and active regulation of cellular processes. The regulation of barrier permeability can be modified by various physiological and pathological conditions, allowing controlled access for certain therapeutic agents while maintaining protection against harmful substances. Some regions of the brain have naturally more permeable barriers, allowing specialized functions like hormone release or chemoreception that require direct contact between neural tissue and circulating substances. The cerebrospinal fluid that bathes your brain and spinal cord serves multiple functions beyond simple cushioning and protection. This clear, colorless fluid actively circulates through specialized pathways, carrying nutrients to neural tissue and removing waste products. The production of cerebrospinal fluid by specialized cells in the choroid plexus is carefully regulated to maintain appropriate volume and composition. The circulation of cerebrospinal fluid through brain tissue involves complex flow patterns that help distribute nutrients and remove metabolic waste. During sleep, the increased flow of this fluid through brain tissue becomes particularly important for clearing away the cellular debris that accumulates during waking hours. The coordination between cerebrospinal fluid flow and sleep cycles demonstrates another example of the temporal organization that optimizes brain function. The composition of cerebrospinal fluid is actively regulated to provide optimal conditions for neural function. Ion concentrations, pH levels, and protein content are all maintained within narrow ranges that support proper neural signaling. The selective transport systems that control cerebrospinal fluid composition can respond to changing brain conditions by adjusting the levels of various substances as needed. The pressure of cerebrospinal fluid is carefully balanced to provide mechanical protection for brain tissue without compressing delicate neural structures. Specialized drainage systems can adjust the rate of fluid removal to maintain appropriate pressure levels, while production rates can be modified to match changing demands. The regulation of cerebrospinal fluid pressure involves complex feedback systems that monitor multiple aspects of brain physiology. Your neural tissue also demonstrates remarkable abilities to maintain function despite the constant thermal fluctuations that occur with changing metabolic activity and environmental conditions. The heat generated by neural activity must be efficiently dissipated to prevent harmful temperature increases, while mechanisms must also exist to maintain function during periods of cooling. The thermal regulation systems in brain tissue operate with remarkable precision to maintain optimal temperatures for neural function. The heat capacity and thermal conductivity of neural tissue are optimized to provide stable thermal conditions while allowing for rapid heat dissipation when needed. The high water content of brain tissue helps buffer against temperature changes, while the extensive vascular network provides efficient pathways for heat removal. The thermal properties of myelin and other specialized neural structures contribute to the overall thermal management of neural tissue. The molecular mechanisms that sense and respond to temperature changes in neural tissue involve specialized proteins that can detect even small thermal fluctuations. Temperature-sensitive ion channels can adjust their activity based on local thermal conditions, while enzyme systems can modify their function to compensate for temperature-dependent changes in reaction rates. These thermal adaptation mechanisms help maintain consistent neural function across a range of temperature conditions. The coordination between neural temperature regulation and whole-body thermal homeostasis involves complex interactions between brain temperature sensors and systemic temperature control mechanisms. The brain's temperature regulation systems can influence behavioral responses like seeking warmer or cooler environments, while also coordinating with autonomic systems that control heat production and dissipation throughout the body. As you continue to rest in this gentle exploration of neural sophistication, your own brain's temperature regulation systems are quietly preparing for the thermal changes that accompany sleep. The slight cooling that occurs during rest helps optimize the cellular processes that support neural restoration and repair. Your blood vessels are adjusting their diameter to maintain optimal flow patterns, while your cerebrospinal fluid circulation is beginning to shift toward the enhanced flow patterns that characterize sleep. The molecular clocks within your neurons are continuing their precise timekeeping, coordinating the cellular rhythms that will guide your transition into deeper states of rest. These microscopic chronometers operate with the reliability of Swiss watches, yet they're made entirely of biological molecules that assemble themselves and maintain their function through the elegant chemistry of life. Each tick of these molecular clocks brings subtle changes in gene expression, protein synthesis, and cellular activity that prepare your neural networks for the restorative processes that occur during sleep. The beauty of these neural rhythms lies in their gentle inevitability. Your brain doesn't force itself towards sleep through conscious effort or struggle. Instead, it follows the natural cycles that have been refined through millions of years of evolution, allowing the intrinsic properties of neural tissue to guide the transition from wakefulness to rest. Like a river flowing toward the sea, your neural activity naturally moves toward the patterns that support sleep, restoration, and the quiet miracles of cellular renewal that will unfold throughout the night. The symphony of cellular activities that creates this transition operates on multiple timescales simultaneously. Rapid electrical events measured in milliseconds coordinate with slower chemical processes that unfold over seconds, minutes, and hours. The integration of these different temporal scales creates a seamless transition that feels effortless despite its underlying complexity. Your neurons accomplish this remarkable coordination not through conscious control, but through the natural expression of their evolved properties and interconnected relationships. The molecular machinery that supports neural function continues its essential work, even as consciousness fades and sleep approaches. Ion pumps maintain electrical gradients across cellular membranes. Protein synthesis continues in cellular factories scattered throughout neural tissue. Quality control systems monitor cellular health and initiate repairs when needed. All of these processes operate with the quiet efficiency of biological systems that have been perfected through countless generations of evolutionary refinement. The elegance of neural organization becomes particularly apparent during the transition to sleep, when the complex, chaotic activity patterns of waking consciousness give way to the more organized, rhythmic oscillations that characterize different sleep stages. This transformation reveals the remarkable flexibility of neural networks, their ability to support radically different types of information processing while maintaining the continuity of cellular function and structural integrity. The coordination between different brain regions during this transition demonstrates the sophisticated communication networks that link neural systems throughout your brain. Areas involved in arousal and attention gradually reduce their activity, while regions supporting sleep and restoration become more active. The timing of these changes is carefully orchestrated to ensure smooth transitions between different brain states without disrupting essential functions. The chemical environment within your neural tissue shifts subtly as sleep approaches, with changes in neurotransmitter levels and other signaling molecules that support the transition to rest. Adenosine, which accumulates during waking hours and promotes sleep pressure, begins to influence neural activity patterns. Melatonin, released from your pineal gland, helps coordinate circadian timing signals throughout your brain. These chemical changes work in concert with electrical and structural modifications to create the optimal conditions for restorative sleep. The cellular processes that support neural restoration become increasingly active as sleep deepens. Protein synthesis increases to replace worn cellular components. DNA repair mechanisms become more efficient at fixing genetic damage. Waste clearance systems operate at peak efficiency to remove metabolic byproducts. The coordination of these restorative processes with sleep cycles ensures that neural maintenance occurs when it will least interfere with conscious brain function. Your neurons also begin to replay and consolidate the patterns of activity that occurred during the day, a process that helps integrate new information with existing knowledge and strengthen important memories. The replay of daily experiences during sleep involves precise coordination between different brain regions, with patterns of activity that occurred during learning being reactivated in specific sequences that promote memory consolidation. The molecular mechanisms underlying memory consolidation during sleep involve complex cascades of gene expression and protein synthesis that can create lasting changes in synaptic strength and connectivity. The timing of these molecular events is carefully synchronized with sleep cycles to optimize memory processing while minimizing interference with other essential functions. The precision of this timing demonstrates once again the remarkable sophistication of neural organization and the importance of sleep for optimal brain function. As your own neural networks continue their gentle transition towards sleep, they're participating in one of the most ancient and essential biological rhythms on Earth. The alternation between activity and rest, between information processing and cellular restoration, represents a fundamental pattern that has shaped the evolution of complex nervous systems for hundreds of millions of years. Your brain's ability to seamlessly transition between these different states reflects the deep evolutionary wisdom embedded in neural architecture. The quiet transformation now occurring within your neural tissue connects you to every sleeping brain throughout history, from the simplest nervous systems that first evolved rhythmic activity patterns to the most complex networks that have ever existed. The molecules and mechanisms that support your transition to sleep are variations on themes that have been refined across countless generations, creating systems of extraordinary reliability and elegance. The peace that accompanies this neural transition reflects not just the absence of active stimulation, but the positive presence of restorative processes that will maintain and enhance brain function throughout the night. Your neurons are not simply shutting down. They're shifting into different modes of operation that support cellular health, memory consolidation, and preparation for another day of conscious experience. This transformation represents one of the most beautiful examples of biological sophistication. Systems that can maintain their essential functions while regularly cycling through dramatically different states of organization and activity. The gentle transition your neurons undergo each night represents more than a simple shift from wakefulness to sleep. It's a transformation that reveals the profound interconnectedness between individual cellular function and the larger rhythms that govern all life on Earth. As your neural networks settle into their nighttime patterns, they're synchronizing not just with each other, but with cycles that stretch back to the earliest days of life itself, when simple organisms first learned to coordinate their activities with the rising and setting of the sun. The molecular conversations occurring within your resting brain involve languages far older than human speech. Chemical messengers that first evolved in ancient single-celled organisms continue to carry information between your neurons. Their molecular structures, refined but fundamentally unchanged across billions of years of evolution. These ancient communication systems operate with a reliability that human technology can barely match, transmitting crucial information with precision that ensures the continuity of consciousness across the daily cycle of sleep and waking. Your brain's electrical activity during this transition creates patterns that would be recognizable to any neuroscientist studying neural function, yet each pattern is as unique as your fingerprint. The specific rhythms generated by your neural networks reflect the accumulated experiences of your lifetime, the particular connections that have been strengthened through learning, and the individual variations in cellular properties that make your brain distinctly yours. This personalized neural signature emerges from the interaction of universal biological principles with the unique circumstances of your individual development. The energy conservation that occurs during sleep represents one of evolution's most elegant solutions to the challenge of maintaining complex neural networks. Rather than simply reducing activity across all brain systems equally, your neurons selectively modify their function in ways that preserve essential processes while allowing others to temporarily decrease. This selective modulation requires sophisticated coordination between different brain regions, each adjusting its activity level based on ongoing communication with the rest of the nervous system. The metabolic shifts that accompany sleep involve changes in how your neurons process glucose and other energy sources. During wakefulness, neural tissue operates with high metabolic demands that require constant glucose delivery from the bloodstream. As sleep deepens, some brain regions can shift to alternative metabolic pathways that are more efficient for certain types of cellular maintenance. This metabolic flexibility allows your brain to optimize energy utilization for the specific demands of different sleep stages. The protein synthesis that increases during sleep involves the production of thousands of different molecules, each designed for specific cellular functions. Structural proteins help maintain the physical integrity of neural connections. Enzyme proteins facilitate the biochemical reactions that support cellular metabolism. Transport proteins move essential substances between different cellular compartments. The coordination of this massive protein production effort requires precise timing and resource allocation that occurs automatically without conscious awareness or control. The dream states that emerge during certain phases of sleep represent some of the most mysterious aspects of neural function. The brain regions that generate dreams can create vivid sensory experiences, complex narratives, and emotional responses that feel completely real despite occurring in the absence of external stimulation. The neural mechanisms that support dreaming involve the activation of sensory and memory systems in patterns that would normally require actual environmental input, demonstrating the remarkable capacity of neural networks to generate internally driven experiences. The memory processing that occurs during dreams may serve important functions in organizing and integrating the vast amounts of information encountered during waking hours. The seemingly random combinations of memories and experiences that characterize dreams might actually reflect systematic processes that help identify meaningful patterns and connections between different types of information. The emotional content of dreams may be particularly important for processing experiences that have personal significance or emotional relevance. And now, as these gentle revelations about your neural architecture drift softly through your consciousness, let yourself rest in the quiet knowledge that within your mind lies one of the universe's most extraordinary creations, your neurons, those 86 billion microscopic architects of awareness, continue their essential work, even as your thoughts grow soft and your breathing slows. They are the living proof that complexity and elegance can coexist in perfect harmony, that the most sophisticated networks ever discovered can operate with the gentle reliability of a heartbeat. Sleep well, knowing that your brain will continue its quiet miracles throughout the night, maintaining, restoring, and preparing for tomorrow's symphony of consciousness. Good night.